Hello, and a very warm welcome to today's Nucleic Acid Insights webinar titled Scalable Chromatographic Strategies for mRNA and saRNA Purification from IVT. I'm Caitlin Crouch, and I'll be moderating today's webinar. And joining me today is Tomas Kostelik, who will explore the development of new chromatography tools for the purification of mRNA and saRNA. After our presentation today, we'll have a live Q&A session, so feel free to pose your questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to them during today's session. I'd also like to draw your attention to the Resources tab on the right, where you can find more information on the topics we're going to be covering today. Now I'd like to introduce our presenter. Thomas holds a master's degree in chemistry from University College London. He's been with BIA Separations for over 8 years, where he currently holds the position of product manager for nucleic acid chromatography. His work includes analytics and purification of viruses and nucleic acids, support of process development activities, and for new product development. Before we start today's presentation, we just want to ask our audience a very quick question, which should pop up on your screen now. Great, thank you so much for your input. So without further ado, I'm going to hand you over to Thomas to kick us off with today's presentation.
Thank you, Caitlin, and welcome to everyone from my side as well. It's my pleasure today to be able to share with you some of our experiences with purification of mRNA and purification of self-amplifying messenger RNA, and describe how we're implementing our existing tools and how we're developing new chromatographic tools which can help us increase the productivity and increase the robustness of purification processes for mRNA and self-amplifying RNA. Before I go into the technical aspects of purification of mRNA and saRNA, I just wanted to spend a few moments to highlight an interesting observation when it comes to development of mRNA and saRNA therapeutics over the last few years, specifically during the COVID-19 pandemic. Very early on, we had seen strong development and strong pipeline for both mRNA and saRNA vaccines against SARS-CoV-2. But it's not until last year that we saw a first approved self-amplifying RNA vaccine against COVID-19, whereas we had mRNA vaccines for the past few years by now. Now we need to also highlight that Genova also developed an saRNA vaccine, which was approved for emergency use by the Indian government, but it has not entered into commercial manufacturing. It has not been approved for commercial supply as a vaccine against COVID-19. Now, when we look at the pipeline for mRNA, in general, we can still see that the majority of the pipeline is still in preclinical development, and very few or a small fraction of this pipeline has reached a late-stage clinical trial or commercial supply. As mentioned, there are at the moment 2 marketed vaccines against COVID-19 based on messenger RNA. Now, there are other application areas. One very strong one is, of course, cancer immunotherapy, where mRNA and saRNA are both very strongly represented. But perhaps another one which is in development, and in the case of self-amplifying RNA, mostly in preclinical development, it's the use of saRNA as a protein replacement therapy. And protein replacement therapy can present a different challenge compared to vaccination, because in the case of protein replacement, we will need potentially repeat and frequent dosing on a more regular basis, and this has a completely different requirement in terms of the immune response and immunogenicity of the therapeutic being used for protein replacement. What are the differences in terms of the structure for mRNA and saRNA molecules? On one side, we have the traditional messenger RNA, which in terms of the structure is composed of a 5' cap, a 3' poly-A tail, and our gene of interest with 2 UTR regions with 1 UTR region on either side. And in terms of size, these mRNA therapies, these mRNA molecules typically range from 1 to around 6,000 nucleotides. Now, the primary difference with a self-amplifying RNA molecule is that the self-amplifying RNA molecule contains also genes encoding for replicase. So these encode for an RNA-dependent RNA polymerase, which typically comes from either an alpha virus or a flavivirus, and they give the RNA molecule the ability to reproduce, to copy itself inside of the cell. And by doing so, we can actually achieve a similar therapeutic effect by injecting a smaller amount or a smaller dose of the RNA molecule. To contrast, the currently approved therapies for mRNA would be in the range of 30 to 100 micrograms RNA per dose, whereas self-amplifying RNA therapies would typically range between 1 and 10 micrograms RNA per dose. And so this ability to replicate on one side allows us to have a smaller dose. On the other side, it also allows us to have potentially a reduced immunogenic effect, allowing us to have a more repeat administration, which we will see the development potentially going towards protein replacement therapies and similar areas. Both mRNA and self-amplifying RNA are produced by in vitro transcriptional reactions, which are cell-free processes where we combine a series of reagents, which include a DNA template, typically a plasmid DNA or a PCR DNA template, with enzymes, which will transcribe the DNA into RNA, and the building blocks and other reagents needed for this reaction to proceed. So this is a cell-free process, but even despite being a cell-free process, we have approximately 14 or 15 different components in an IVT reaction, and the ratio and the interaction between all of them is very important to achieving a high purity and high yield in the IVT process. So cell-free and synthetic does not translate to simplicity of the IVT reaction, but this still requires extensive optimization for different constructs. Now, the reaction itself is a very fast process, typically takes 2 to 3 hours to complete when we're to finish when we're producing RNA in a batch IVT process, and they are also very high yielding IVT reactions. So we can use a relatively small volume of the reaction mixture to produce a relatively large number of doses of RNA. And these two really make mRNA and saRNA attractive to use in rapid responses, for example, during pandemics of emerging viruses. Now, the speed of the reaction and the high yield are also two challenges for analytics. Because of the speed of the reaction, we need to have very fast analytical methods to be able to keep track and monitor these processes in real time. And the high yield is a stark contrast to fermentation processes, where typically, we would have, in a fermentation, we would have a large amount of contaminants with a relatively small amount of our product. In the case of RNA, we have a small amount of contaminants in a large amount of our drug substance, of our RNA product. And so this means that the analytical methods need to provide sufficient sensitivity to detect all of those contaminants. Now, we've talked about the structure of RNA, which consists or contains the 5' cap and the poly A tail. These can be added onto the structure of RNA in a single pot reaction. So we can have a DNA template which encodes the poly A tail, and we can include a cap analog in our IVT reaction, which will give us the mature RNA in a single step process. Alternatively, we can look at adding the 5' cap and the poly A tail in secondary reactions. So we would have an initial IVT, which just produces the RNA construct, and then we would have a tailing reaction and capping reaction separately. Now, all of these raw materials and reagents used in an IVT reaction also bring contaminants along from their own production processes. So when we think about purification of mRNA, we need to think about not just the RNA product itself, but also the raw material contaminants introduced into the IVT reaction from the different reagents, the reagents themselves, so the DNA templates, the nucleotides, the enzymes, they need to be separated and removed from our RNA product. And then we need to look at product-related impurities, which consist of truncated RNAs, fragments, double-stranded RNA, which chemically speaking are the most similar to our RNA product, and also some of the more challenging to remove in many cases. We need to keep in mind that RNA is a labeled molecule, so when we process it and when we select how to purify it, we need to use methods which will not degrade or result in loss of our RNA product. Think about purification. Typically in an industrial setting, chromatography is a very well accepted tool, a very well accepted method to purify different types of therapeutics, thanks to its scalability. So we can purify bench scale all the way to commercial manufacturing scale, and we can also achieve a good purity, so a good separation from products and contaminants. Now, in the workflow of mRNA production, there is actually a lot of place for chromatography as well. On one side, the different analytical aspects of the RNA process, starting from looking at the quality and the purity of the raw materials, analyzing and understanding the quality of the plasmid entering our IVT process, as well as the remaining reagents or chemicals which are used in this reaction, which will allow us to then ensure a high quality and high purity of the IVT product. Once we have these raw materials selected, we then go into the IVT reaction itself, which, as mentioned earlier, is a very fast process and one that contains a whole, a large number of reagents, which we need to understand and optimize to get the best performance of the IVT reaction. And this one is particularly important because the cost of producing RNA is typically very high, and this optimization of the IVT reaction is one of the key areas that can help us reduce the production cost of RNA therapeutics. Once we produce the RNA, we can of course look at chromatography for purification, so as a unit operation in the downstream process, but we can also look at analytical chromatography to support us with optimization of the purification process, so to optimize the chromatographic steps, to optimize the filtration, the TFF steps. But also we can use then analytical chromatography to monitor and track in real time the purification process of our RNA product. And so we can use analytical chromatography as an in-process analytical tool, which will help us ensure that we get a consistent quality and a robust production and purification process. Finally, once we have purified our drug substance, we of course need to understand the stability and the quality of the drug substance. So these are two important areas where analytical chromatography plays an important role. And chromatography was first developed over 100 years ago, when the types of therapeutics, the types of molecules that were purified were very different from what we're working with nowadays with mRNA and self-amplifying RNA. Traditional chromatography was developed more than 100 years ago, when the size of the particles of the molecules that were purified ranged, from aspirins, from small molecules to things like small globular proteins, something like a monoclonal antibody. When we compare that to messenger RNA or to plasmid DNA and other large biotherapeutics, we can see that we have an order of magnitude difference in terms of the mass of the RNA of the particle. But we can also see that the size and shape, of these molecules is no longer comparable. So a protein would typically have some kind of globular shape, but when we look at mRNA, this is a long, stringy molecule with a lot of different branches for its secondary structure. And so if we lay down this molecule flat on a surface, it would be in the range of hundreds of nanometers in size. Similar for the delivery vehicle, which is commonly used for mRNA delivery, lipid nanoparticles typically are 1 or 2 orders of magnitude larger than the small globular proteins. And so chromatography was initially developed for these small molecules, which in turn brings challenges when we bring the same technology over to purification of large messenger RNA. Now, traditionally, porous particle columns consist of a resin with chromatographic beads with a porous surface, and these provide a very good chromatographic performance in terms of binding capacity, productivity for small molecules and proteins. As we increase the size of the particles we're purifying, and here we're talking about DNA plasmids, which range in size from hundreds of nanometers in size to even micrometer size, the performance of these porous particle columns decreases very rapidly. So as we get into DNA template, as we get into RNA purification, a different type of chromatographic stationary phase is needed to achieve a high productivity and a high recovery for these molecules. And the structure of a chromatographic monolith is very different compared to a porous particle resin. The monolith was developed, first of all, to be made up of a single block of polymer. So unpacking, opening one of these chromatographic columns would show to us, a solid stationary phase, which is where the word monolith is derived from. And if we zoom in, if we look into the microstructure of these stationary phases, we would see that the surface does not consist of dead-end pores, but instead the surface consists of large flow-through channels. Now, these channels provide us with, first of all, unobstructed flow for the large size of the molecules we're purifying. The channels themselves range in size from 1 to 6 micrometers in diameter, with distinct sizes determined based on the molecules that we're purifying. But the large channels also give us the advantage of having a low operating pressure. So with this low operating pressure combined with a good surface accessibility for the molecules we're purifying, we can accelerate, we can intensify the purification of these large biomolecules. So all of the material, all of the RNA or DNA flowing through the structure of these channels will be in direct contact, in direct access to the entire binding surface area, which means that we eliminate the process of diffusing our RNA, our DNA into these dead-end pores. Instead, we have a process entirely based on convection, which allows us to have flow-independent capacity and flow-independent performance of a chromatographic column. So when we start to look at analytical chromatographic methods that we use in the mRNA production and purification workflow, I can illustrate here two different approaches that we use very extensively. One will be perhaps more familiar to anyone coming from a monoclonal field, and the other one will provide a bigger or improved insight into what is happening in the IVT reaction. On the left-hand side here, we can see an analytical method based on affinity chromatography, which relies on the ability or the presence of a poly-A tail on an mRNA construct and binds or catches the RNA with the help of an oligo-DT ligand. And so this column, when we inject a sample, whether it is crude IVT or a purified RNA sample, will allow us to detect and quantify the concentration of RNA in any sample in a very short time, in a very short duration. This method is very akin to the protein A ligands in the monoclonal antibody field. So it provides us with a fast analytical method with only one caveat: the fact that it gives us only a single parameter, only a single output for the analysis. Now, when we talk about the IVT reaction, we saw earlier that it contains a lot of different building blocks which are critical in the production of RNA, because without the building blocks, we can not produce RNA anymore, and our reaction will stop. And so we have developed and optimized this multimodal chromatographic ligand called Prima S, and we packaged it into this CMAC analytical column format, which allowed us to develop an analytical method to separate and quantify the key different building blocks in the IVT reaction. So if we look at the chromatogram here, which is an overlay of the different IVT components, we can see that the analytical method will allow us to quantify the RNA concentration, and it will also allow us to detect the DNA templates, to quantify the different nucleotides in the reaction, and to quantify the amount of capping reagent present in the IVT reaction. Now, with all of this information, we can get a much better insight into what is happening in the IVT process, and we can use this method to optimize IVT reactions. The method is very fast, and in a time of 3 minutes per sample, we can get a very clear picture of the key building blocks present in our IVT process. The way we use it typically is by taking 1 to 2 microliters of sample every 20-30 minutes from an IVT reaction, analyzing it on the analytical column, and obtaining a data point which corresponds to the amount of RNA being produced and the amount of different nucleotides remaining in the IVT reaction. Now, by monitoring this, we can get a very good picture on the mRNA production kinetics, and we can identify the conditions, the IVT conditions, which give us the maximum productivity for our certain specific construct. Because the method is so fast, we can actually use this to monitor 5 or 6 different IVT reactions in parallel, simply by injecting RNA onto an analytical system and obtaining this information for every single reaction individually. We can also keep very good track of the reaction, of the consumption of nucleotides and the concentration of capping reagent remaining in our IVT reaction. Now, with this type of tool, we can gain a much better understanding of how our IVT reaction is working, and we can also look at developing a feeding method where we can potentially supplement or add additional nucleotides into the IVT reaction as these are being consumed in the production of mRNA. This is an approach that we have published 2 years ago in Chemie Engineer Technique, where we have shown that we can optimize a reaction in a thermal shaker, establish or characterize very effectively the consumption of nucleotides, and develop a model where we can spike or feed additional nucleotides into the IVT reaction as these are depleted in our existing reaction vessel. And by doing so, we can extend the duration, or we can prolong the duration of the IVT reaction, which allows us to make much better use of all of the remaining reagents, including the capping reagent, including the enzymes which are present in our IVT reaction. Now, this type of addition can be done manually, as is shown on the slide here, but we can also develop a model for the rate of consumption of the nucleotides. We can then feed this model into an automated system, for example, the Ember 250, which allows us to feed with a specified program the nucleotides into the IVT reaction. And the result of scaling up a reaction on such a device, we can see that we initiate the reaction. Once the concentration of nucleotides falls below a certain threshold value, we initiate the feeding mechanism, which allows our IVT reaction to continue producing RNA without interruption. In this case, we've stopped the reaction after 7 hours, which was the longest we could do in a typical working day. And we have achieved a final concentration of RNA in this IVT process of 12 grams per liter. Now we can see on the graph that the concentration of RNA is not increasing at the same rate throughout the entire reaction. Here we need to keep in mind that as we feed nucleotides, we're also adding additional volume into the IVT reaction. So the concentration of 10 milligrams per milliliter after 4 hours of reaction time and the concentration of 10 milligrams per milliliter after 6 hours reaction time actually has a much higher yield at the later time points. Now, another way to look at this is to look at the transcription factor, so the rate of production, the mass of RNA produced per mass of pDNA used as a template in the reaction. And we can see that comparing the reaction tube and the Ember, so the scaled up reaction, we can see that the rate of production of RNA is kept consistent. So with the help of this Prima S method, we can achieve not just optimize the parameters going into the IVT reaction, but we can also develop a specific feeding mechanism to achieve in the future continuous RNA production in IVT processes. The RNA that we produce in the IVT reaction then, of course, needs to be purified. And one of the more common methods to do so is the use of affinity chromatography by simultaneous oligo-DT. Now, this method does not need to be introduced in detail, but there are some aspects which become relevant when we talk about mRNA and SA-RNA differences. RNA is bound on the oligo-DT column by a moderate to high level of salt. The presence of salt allows the formation of base pairs between the poly-A tail of the mRNA molecule and the oligo-DT ligand. Non-polyadenylated constructs and reagents will flow through the column without binding. We then typically perform a low salt wash step, which helps reduce the conductivity of the solution ahead of an elution step, which we normally perform in water, which allows us to recover, in most cases, 90 to 100% of the RNA loaded on the column. One of the key aspects that we always strive to when we develop purification of mRNA is to design a purification process with the minimal holding time between different unit operations and avoiding unnecessary unneeded steps in the process, like freezing and thawing and implementation of diafiltration and ultrafiltration steps before chromatography. The most optimal workflow we have found was to take an IVT reaction, which we generally quench with some kind of EDTA containing buffer, followed directly by capture on chromatographic monoliths directly from this mixture by adding a moderate to high level of salt, as mentioned before. So what we would like to do is to achieve a process which is continuous and uninterrupted from the IVT reaction to the purified product, purified drug substance after the oligo-DT chromatographic step. With the help of the simultaneous oligo-DT, we can do this in a very short residence time on the column, thanks to the high flow rates and high binding capacities that we can achieve with the monolithic structure of the chromatographic columns. To achieve the best chromatographic performance from the oligo-DT or from any chromatographic step, we typically implement a design of experiment approach where we evaluate different binding and sometimes different elution conditions on a 96 well plate format, which contains small amounts of the same monolith that is found then in the larger scale-up chromatographic column. Now, in this study that we have published for oligo-DT, we've shown that using guanidine hydrochloride and actually magnesium chloride as well have both a significant contribution towards the binding capacity of the oligo-DT monolith. And so we have shown that optimizing the conditions allowed us to go from for one construct to go from a binding capacity of 4 milligrams per milliliter in sodium chloride up to 6 milligrams per milliliter in an optimized buffer composition. And this in turn allowed us to decrease the size of the column needed for purification of RNA by a margin of 50%, which in turn also reduces the cost of production of the RNA. So how does chromatographic purification of self-amplifying RNA differ from purification of messenger RNA? We need to keep in mind some of the properties and structural and chemical properties of these RNA constructs. On one side, we have the larger size of self-amplifying RNA, which gives it a lower stability to external forces like shear forces, but also to lower stability in processing conditions. And we talked about oligonucleotide chromatography, where we use elevated salt concentrations. The same concentration of salt will not be applicable to purification of a small mRNA molecule compared to a large self-amplifying RNA molecule. So we need to optimize and design the process to fit the needs of the self-amplifying RNA construct. Another aspect to keep in mind is the size or the length of the self-amplifying RNA construct. When we talk about binding on oligo-DT, the poly-A tail is the key part which allows us to purify mRNA. And in self-amplifying RNA molecules, this poly-A tail is less than 2% of the full length of the RNA molecule. And this means that combined with the complex secondary structure, this may result in poor accessibility of the poly-A tail, which would prevent or reduce the ability of RNA constructs to bind onto the oligo-DT column. We need to keep in mind that the secondary structures of RNA are also dynamic. They vary not just between individual molecules, but they can vary also when the molecule is exposed to different solutions, comparing to, for example, going from high salt to low salt environment. And finally, the one thing to keep in mind as well for purification of self-amplifying RNA is that the size, the footprint, the size of the molecule increases more rapidly than the mass of the molecule itself. And in turn, if we take a chromatographic monolith or a chromatographic media, and we cover the surface with self-amplifying RNA, and we cover the surface with a smaller standard RNA molecule, we would typically expect that the smaller mass of self-amplifying RNA would be bound to the same surface area. In other words, we would typically see a lower binding capacity for self-amplifying RNA. So how can we look, or how can we solve some of these challenges? We cannot change the chemistry or the structure of self-amplifying RNA, but we can change the way that we process and the way that we purify these self-amplifying RNA constructs. Now, the first one is looking at oligo-DT chromatography, where we use these elevated salt concentrations, which can be potentially challenging to the stability of RNA constructs. We also need to remember that salt concentration has a direct correlation with the binding capacity for a certain construct on oligo-DT chromatographic media. So optimizing the salt concentration has to be done towards the highest possible salt concentration, which will give us the highest possible capacity, but remaining below the threshold concentration when we start to see precipitation of RNA. And one way to overcome or to reduce the chances of precipitation of RNA is to implement an approach called inline dilution. This is a method which is very frequently used in industrial production and purification of biologics, but not yet or not so frequently implemented in RNA purification. A traditional, I would say, batch processing of RNA would typically be, taking the IVT reaction, quenching it with EDTA, where we have our RNA product in a relatively stable condition, and then dilution or addition of a binding buffer which contains sodium chloride. Now, addition of this salt to the sample reduces or increases the risk of precipitation of RNA, and beyond this point, any holding time, any process holding times before the material reaches the chromatographic column can result in potential precipitation. And this would then have a negative impact not just on loss of the material, but it would also require additional filtration steps before the sample can be applied on chromatographic columns. This is really easy to overcome with the help of inline dilution, where we follow the same initial procedure of quenching the IVT reaction, and we keep our RNA product in conditions that we evaluate and test that provide a stable environment for the RNA, and we add the high sodium chloride concentration or the high salt concentration by a method of inline dilution. So on our chromatographic system, we have on one pump the IVT, the quenched IVT reaction, and on the other pump a 2 times dilution buffer or a binding buffer, which we then apply in a gradient mixing approach, where our IVT reaction will be mixed with the dilution buffer and reach the column with only seconds of contact time, or not even seconds of contact time between the high salt and the RNA sample. And this reduces this contact time with the high salt, allowing us to have better stability of the RNA and robustness of the RNA process. Another way to overcome stability concerns is the use of chromatographic methods which do not require elevated salt concentrations for binding. And one of the first thoughts comes to mind is the use of ion exchange chromatographic media, which can bind the phosphate backbone of the RNA, which has a negative charge throughout the entire length of the molecule. The issue that we face here is that when we take traditional ion exchange chromatographic media, they exhibit very strong binding of messenger RNA, both mRNA and saRNA, and this results in lower recovery, so lower elution recoveries, and may and typically do require elevated temperature to achieve complete elution of the RNA molecule. Now, this is true for both strong ion exchange media like QA, for example, which is consistently charged, positively charged throughout the entire pH range, but also for weaker anion exchange media, for example, a very common one, diethylamine, which does lose the charge beyond the pH of 10.5, and is uncharged above a pH of 11 or 12. Now, this one may elute the RNA in potentially in room temperature, but it would do so at a very high pH, where the stability of the RNA would be a concern. And so we have looked on our side at over the last years at different types of ligands that combine an ion exchange character with the ability to form hydrogen bonds, which are very extensively, and the ability of hydrogen bonds is very extensively present in all biomolecules, including messenger RNA and including DNA. So the molecule of mRNA has both, this negative charge, which would effectively interact with the weak ion exchange character of the multimodal ligand, but it also has this ability to form all of the different hydrogen bonds throughout all of the bases of the RNA molecule. So we can take a ligand, and this is the same ligand we saw earlier for analytical purposes, which loses its charge at a lower pH value. So the isoelectric point is at a pH of 9.5, and it loses its charge at a pH of 10.5. And this allows us to effectively capture and purify single-strand mRNA on a multimodal column and eluting it at room temperature. The workflow that we would typically implement for this purpose would be to take the IVT mixture exactly the same way as with oligo-TT. We quench the IVT mixture with addition of EDTA, and we then perform a dilution with a binding buffer, which in this case does not contain any salt, but rather is pH 7 or pH 8 buffer, which allows us to bind the RNA on the column. Now, this Prima S column has a very different selectivity for DNA, so for the template compared to the RNA molecule. And so we can perform a high salt wash step, typically with 1 molar sodium chloride, which will elute all of the DNA template. It will also elute different enzymes and unincorporated nucleotides, and keep our RNA bound on the column. So the RNA does not elute even in 1 molar sodium chloride. So to elute the RNA, we can then perform a pH step or a pH gradient, which allows us to elute the RNA from the column in a tight, highly concentrated peak. Now, the pH for elution in the case of Prima S is a pH of around 10.5. So we need to neutralize the eluate immediately with a concentrated neutralization solution, which allows us to produce a workflow or a purification method, which ensures that we have a stable elution product. Now, with the help of this multimodal chromatography, we can bind a very large amount or a higher amount of RNA compared to an oligo-TT workflow. For example, we have shown that with the help of Prima S, we can bind up to 7 or above 7 milligrams of RNA per milliliter of chromatographic column. So the question that we then needed to answer was, can we find a chromatographic ligand which can elute the RNA at a lower pH value, but still retain the good selectivity between the RNA molecule and the DNA template and the proteins and enzymes and so on, which are present in the IVT reaction? And this is how we've identified this multimodal ligand, which we then called Swiper, which allows us to follow a very practically the same workflow as with Prima S, where we take the IVT reaction, we dilute it with a binding, but we quench it and we dilute it with a binding buffer, and we load it directly onto the column itself. Now, in this case, we use a pH of 5 to 6 to achieve binding of the RNA on the column. We follow the same high salt wash step to remove templates to remove other contaminants. And then we can do, again, a step or linear pH gradient elution to recover the RNA product. Now, the key difference here is that we achieve elution of the RNA at a pH of 7 to 7.5, which means that upon elution, the RNA does not need to be neutralized and is actually present in a buffer, which can frequently used for other downstream applications. Both Prima S and Swiper therefore give us a chromatographic approach which does not rely on high salt concentration for binding, and this has a strong advantage for purification of messenger of self-amplifying RNA. Now, with Prima S, we have seen that self-amplifying RNA can be retained also very strongly on the column, requiring a higher elution pH to elute the RNA from the column. But in the case of Swiper, we have seen that we get a very good, very consistent performance for RNA regardless of size, going from a small, let's say 1,000 nucleotide eGFP mRNA, all the way up to a 10,000 nucleotide self-amplifying RNA. We can clearly see on the chromatograms that the resolution, the separation between the DNA template and the RNA molecule is maintained consistent regardless of the size of the construct. And we have seen that even for self-amplifying RNA, the recoveries would be above 80%, with recoveries reaching 100% for smaller RNA constructs. And the NTP, the DNA template, the enzymes are typically removed in the flow-through or in the high salt wash step in this purification process. With oligo-DT, with Prima S, and with Swiper, we've seen that the elution conditions are typically in some kind of low salt or low conductivity type of buffer. So in the case of oligo-DT, we generally elute with water. In the case of Swiper and Prima S, we typically elute in using pH, so we do not need to increase the conductivity for elution of the RNA molecule. And this gives us the ability to apply these three different columns to perform desalting of RNA samples at any scale, all the way from small scale laboratory work and up to large scale manufacturing. So we can use chromatographic processes to do desalting in a much faster workflow or a much faster fashion compared to the use of, for example, buffer exchange to achieve desalting of a sample. Now, so this was the case with RNA constructs. It is the same with plasmid DNA. We can also develop on Swiper, for example, a workflow to desalt a sample of plasmid DNA, with the only difference being that we need to load or we need to dilute the sample below a certain threshold value where the DNA would elute from the column. But after dilution, we can load it on the monolith, and again, the loading phase on the monolith will be very fast because we can achieve high short residence times and high volumetric flow rates. And so we can load the DNA sample on the column. We can do we can do a low salt wash, and then we can do a pH elution, which would give us a plasmid DNA sample, which is desalted and ready to use in other downstream applications. So all of these columns, they provide us not just the ability to use for purification of RNA, but also the ability to do desalting, which can apply more broadly to different types of RNA molecules or biologic molecules as well. So throughout this webinar, we have talked about the different ways we can capture messenger RNA and self-amplifying RNA from an IVT reaction mixture, focusing, of course, on the differences between purification of self-amplifying RNA and purification of messenger RNA in the case of these different chromatographic media. There is then, of course, an entire different world, which is the polishing of messenger RNA, where we would typically employ different chromatographic monoliths to reduce specific contaminants like double-strand RNA, like endotoxins, like protein contamination, and so on. But this would be, of course, a topic for a completely different. Now, with that, I would like to just bring up some of the key points that were presented. And so, we've looked in detail about the differences between messenger RNA and self-amplifying RNA in terms of the construct and how it affects the chromatographic purification across the different known types of chromatographic media. Specifically with oligo-DT chromatography, the binding conditions on one side need to be optimized individually for different constructs to achieve the best process performance. And on the other side, we've talked about how the elevated salt concentration can be more challenging in the case of purification of self-amplifying RNA. And we've looked then at how we can use different methods to overcome these challenges. So the first one would be to use inline dilution, which reduces the contact time between the high salt concentration and the mRNA construct, And this approach is very favorable, especially with RNA constructs which are more prone to precipitation when exposed to even moderate levels of concentrations of salt. Now, to avoid altogether the use of high salt concentration for binding, we can use a multimodal approach. For example, with Prima S, which we discussed both from the analytical perspective and from the purification perspective, we can bind RNA in a neutral pH. The same with the Swiper molecule, the Swiper chromatographic column. And with these columns, we can also achieve elution of RNA at room temperature. In the case of Swiper, we do this in a neutral pH. So when we talk about the stability and the purity of the RNA, especially for self-amplifying RNA, the use of the Swiper chromatographic column would provide us with the ability to purify by maintaining gentle purification conditions from the IVT reaction all the way to the purified RNA drug substance. With this, I would like to finish off and invite everyone who would like to learn more about RNA purification to attend the 10th Monolithic Symposium, which takes place in June on June 3 to 7 in Slovenia, and there will be talks from industry experts discussing purification of not just nucleic acids but also other biological constructs used in gene therapy applications. I would also like to thank everyone from the team in Slovenia who are working tirelessly day and night to develop purification processes for all of these different constructs and molecules, as well as, of course, the R&D and development teams who are developing all of these wonderful new chromatographic columns. And with this, I would like to everyone for listening in and welcome all of the questions that you may have.
Great. Thank you so much for a great presentation, Thomas. You're welcome to stop sharing your screen and then you can pop on your camera and join me for the Q&A. Fantastic. So before we start our Q&A today, we just have one quick question we'd like to ask the audience, which should pop up on your screens now. Great, fantastic. So we've got a few minutes to do a quick Q&A, Thomas. So I'm just going to dive straight into it. Our first question here asked by an audience member is, what is the expected binding capacity of oligo-DT for self-amplifying mRNA?
Yeah, so as mentioned, during the webinar, when it comes to self-amplifying RNA in general, because of the larger size and footprint that it would occupy on a stationary phase, the binding capacity would be lower compared to messenger RNA. So if we talk about self-amplifying RNA specifically, we would tend to detect or see binding capacities in the low milligrams of RNA per milliliter of the stationary phase. This, of course, is a little bit construct dependent, is also dependent on the optimization of the binding conditions. And again, here it's a place where this inline dilution can really help, boost the binding capacity also for these different self-amplifying RNA constructs on the oligo-DT medium.
Great. So our next question here asks, is there any entrapment of self-amplifying RNA in the channels of the monolith?
This is a fantastic question, and it ties on to what I've mentioned early on at the end of the presentation. So we have seen that, mRNA tends to be flexible enough, at least inside of the structure of the monolith and in the processing conditions that we use to prevent binding or sticking of the RNA in different parts of the monolithic column. So we have seen very high recoveries for self-amplifying RNA across all of these different tools that I have presented here, and so the binding or entrapment would not be observed or would not be seen or measurable in the case of self-amplifying RNA.
Great, fantastic. So I think we have time for one more quick question. This audience member asks, is it common to use high concentration of organic solvents in manufacturing of RNA? And how are organic solvents removed after SDVB?
Okay, yeah, so we did not touch on this. This would be a topic to discuss on the polishing aspects of RNA, but organic solvents we typically employ when we do purification with reverse phase chromatography, and we see this as a fantastic tool to reduce double-strand RNA contamination in our mRNA products. And so, we would typically use some kind of reverse phase method where we would, elute the RNA with the help of an organic solvent. And then following this, we would do some kind of buffer exchange, which would allow us to remove or reduce or remove the solvent from the sample.
Great, fantastic. Thank you so much for that answer. So unfortunately, that is all we have time for today. Any questions we didn't get to during our Q&A session today, BIA and Thomas will answer offline for you. Just a reminder to look out for an email from us tomorrow, as the on-demand for this webinar will be available from tomorrow. So do look out for that email. And all that's left to do is to, of course, thank Thomas for a great presentation and for a great Q&A. And thank you to the audience for listening. We hope you'll join us again soon.